Good afternoon, everyone. I'm Dr. Rui Chen from Department of Plastic Surgery, Renji Hospital, Shanghai Jiao Tong University School of Medicine. It's a great honor to present our recently study accepted by Nature Communication here in Amsterdam. Now,
Catecholaminergic neurons boost fibroblast osteogenic activity in keloid. Ossified keloids are a highly refractory subtype. They show extreme rigidity and cartilage-like properties that cannot be explained by collagen alone. Strikingly, ectopic mineralization and bone-like foci are often observed. This led to our core question: does a disease-specific osteogenic-like fibroblast subpopulation exist in keloids? Also, keloid patients frequently suffer from itching and pain. Psychological stress itself is an independent risk factor for recurrence. So we asked, do neural signals actively reshape fibroblast fate? We performed ST-RNA-seq on 5 human keloid samples, GT4PS300 cells. We identified the disease-specific mesenchymal fibroblast cluster. GSEA revealed strong enrichment in bone development and neuron projection guidance. Pathways: master osteogenic regulators RUNX2, CoL110, and 1-OGN were highly localized in this cluster. Using RNAscope, we found that RUNX2 IBSP fibroblasts constitute NT5 of total germinal cells in keloid. IBSP protein was deposited in the cytoplasm and paracellular matrix of RUNX2 cells. Histochemical staining showed massive GAG accumulation, increased ALP expression, and calcium deposition. This proves a genuine hybrid fibro-osseous reprogramming in advanced keloid, not just collagen. Build-up. What drives this osteogenic behavior? GCE also showed a strong response to catecholamine stimulus signature with upregulation of GNAS and GMB1. Catecholamines are mainly released by sympathetic nerves. Using 3D whole-mount immunofluorescence for Th tyrosine hydroxylase, we found that normal skin has orderly nerves around hair follicles, but keloid dermis is hyperinnervated by dense disorganized Th fibers. Ex vivo epinephrine or norepinephrine boosted IBSP protein production by 2.58-fold and 17.4-fold, respectively. Thus, aberrant catecholaminergic innervation is a key upstream driver. We screened the entire adrenergic receptor family by qPCR. ADRB1 was selectively and massively upregulated in keloid fibroblasts and colocalized on RUNX2. Cell membranes, knockdown of ADRB1 or treatment with the B1 blocker metoprolol completely abolished. Epinephrine-induced IBSP protein. Surprisingly, epinephrine had zero effect on IBSP mRNA levels, suggesting a post-transcription mechanism. To find the post-transcriptional regulator, we performed bulk RNA-seq and identified translink, TSN, and RN binding protein highly expressed in keloid stroma. Confocal microscopy showed at baseline TSN is nuclear. Epinephrine triggers its rapid nuclear cytoplasmic translocation to form puncta blocked by metoprolol. Functionally, TSN knockdown suppressed epinephrine-induced IBSP protein. Using our endoscope, we visualize that resting IBSP mRNA is trapped in the nucleus. Epinephrine drives its nuclear export. TSN deficiency blocks it inside the nucleus. RAPQ-PCR confirmed that epinephrine increases IBSP mRNA binding to the EIF4E translation complex. Thus, TSN controls IBSP production via mRNA trafficking and translational completed the upstream pathway. Epinephrine ADRB1, CAMPKA, CRB, TSN qPCR proved that CREB1 physically binds to the TSM promoter upon epinephrine stimulation. So the full axis is ADRB1, CAPM, PAC, RB, BTS, NIB. For in vivo validation, we used a rat tail stretching model that mimics human high tension scarring. Systemic 6-ODA chemical sympathectomy led to a 6.5 reduction in scar thickness. It also suppressed collagen, IACE, ASMA, and most importantly, RUNX2 IIBSP expression and GAG accumulation. This proves that catecholaminergic neural input is required for in vivo fibro-osseous reprogramming. Finally, we explored the translational non-invasive strategy. During wound healing and stretching, we gave metoprolol, a selective B1 blocker, in drinking water. Metoprolol significantly reduced scar thickness, suppressed collagen INSMA, and almost eliminated RUNX2 cells, IIBP deposition, and JAG accumulation. This provides strong preclinical evidence for repurposing metoprolol to treat mechanically driven keloid progression. Let me summarize our three main discoveries. First, we identified a distinct RUNX2 IBSP osteogenic fibroblast subpopulation in keloid that drives tissue stiffening and ossification. Second, we elucidated a complete signaling axis, ADRB1, 10PK, ACARBTSN, that controls bone-like matrix synthesis via translation. Third, we demonstrated that the clinically available drug metoprolol can serve as an immediately non-surgical and effective strategy to block keloid progression. Before I give my acknowledgements, let me briefly introduce our institution. Renji Hospital was founded in 1844 by an Englishman named William Lockhart. It was the first hospital practicing Western medicine in Shanghai. Over the past 180 years, it has become a premier academic medical center. Renji has been awarded Shanghai Civilized Unit 16 times and was recently honored as the 5th National Civilized Unit. We are proud of this legacy and committed to advancing clinical research. I would like to express our sincere gratitude to Professor Wang's team at Shanghai General Hospital and the team at the Institute of Molecular Medicine, Shanghai Jiao Tong University School of Medicine, for their invaluable guidance and support. I also thank all members of Renji Hospital who contributed to this work. Thank you for your attention. Wish the conference a complete success.
然后呢？是共享。
So thank you very much. I don't know if you have questions from the audience for you. Can you hear us?
Yes.
Okay, that's good. Any questions?
Yeah. There's a question. Has ever been tried in patients? Kind of indication, easy to get, it's cheap. Very early stage. I mean, Thank you very much for your talk. We're just curious to hear if you have tried it in patients. It's cheap, it's easy to get. We know that it works for hemangioma, so there's some indirect evidence from other diseases, but I'm not aware of any clinical data in real patients so far. Thanks.
非常感谢您的演讲。
This work is the lab work, yes. But the clinical is just follow which medicine is with the most safety to choose. So next work we will try on that.
So did you start a clinical study, or how you're going to tackle this?
Because the medical choose it's not the safe for take the pill just like beta leuker petrol. Yeah.
Okay.
So thank you. As there are no other questions at the moment, I think it's really very important and could be really a way forward. And of course, we are waiting eagerly for clinical data in humans. And maybe another step would still be in an animal model to use this study in further studies. But I think it's really very promising.
And we can use it in our patients tomorrow.
Yes, you could.
Okay, you want to have clinical studies first. Okay,
hesitating a little bit.
Okay, good.
The ethics. Okay, thank you. Thank you very much. Thank you for staying with us and presenting this interesting data.
Yes, okay.
So then we are going to start with the next session, and the first speaker